Yes, uh, let's start the Human Genome Project. Human Genome Project. Yes, what is genome? The entire DNA in the haploid set of chromosome of an organism. The entire DNA in the haploid set of chromosome of an organism, we say genome. In human genome, DNA is packed in 23 different chromosomes. Then Human Genome Project is actually started in 1990, that expected to be completed in 2005, but we are completed earlier in 2003 because of the advancement in the technology. And it's an effort in identifying the sequence of nucleotides and mapping all the genes in human genome. After all, the genome project, we came to identify that human genome contains 3 into 10 to the power of 9 base pairs. 3 into 10 to the power of 9 base pairs. So these calculations to be remembered here: 3 into 10 to the power of 9 base pairs, or we say 3 billion chemical base pairs that make up human DNA. Determine the sequence of the 3 billion chemical base pairs of DNA, human DNA. So that goals of Human Genome Project are identifying all the estimated genes in human DNA, determine the sequences of the 3 billion chemical base pairs that make up human DNA, that storing this information in the databases, improve tools for data analysis, transfer related technologies to other sectors such as industries, that whatever the information we got here, we are going to transfer this particular technology wherever it's necessary, like forensic wing in that particular area, that addressing the ethical, legal, and social issues. In brief, we say ELSI, that may arise from the project. While we are doing this project, whatever the issues arise ethically, legally, and social issues, that addressing all of them is the goals of Human Genome Project. So technical information to be remembered, these 3 billion chemical base pairs from this particular page. Okay, so the Human Genome Project was closely associated with bioinformatics. What is bioinformatics? Application of computer technology and information science to organize, interpret, and predict biological structure and functions. Usually applies in analyzing DNA sequence data. So the methodologies of HGB, that two major approaches we have, that expressed sequential tags, which is most happening question in NEET examination, identifying all genes that are expressed as RNA. That question will come in that particular manner, as earlier in 2019 we got this question, that identifying all genes that are expressed as RNA, that is said to be expressed as sequential tags, ESTs, we use it to see. And the sequence annotation, that sequencing the whole genome which contain all the coding and non-coding sequences, and later assigning different regions in the sequence with functions. Along with the sentence, you need to remember this particular, because in case of Human Genome Project, we have very less technical information. That's why the very limited area, but having enough good scope in NEET examination. So let me say again, identifying all genes that are expressed as RNA. What do you say? Expressed sequential tags. What is sequence annotation? Sequencing the whole genome which contain all the coding and non-coding sequences, and later assigning different regions in the sequence with functions. So, so this is the methodologies of HGP, the two major approaches. These are very important. And let's come to the procedure here. In the procedure of Human Genome Project, we first isolate the total DNA from the cell. We convert them into random fragments. We'll cut them into the random pieces here. For that, we have enough good the technology available, that identifying after the identification of the restriction endonuclease enzymes is became much easier here. That clone these particular DNA fragments, we are going to clone in a suitable host, like either it is bacterial artificial chromosome or yeast artificial chromosome, and with that we do amplification of these particular fragments. That fragments are sequenced using automated DNA sequencers, using Frederick Sanger method, we say. Then these sequences are arranged based on overlapping regions present in them. Sequences are arranged based on overlapping regions present in them. Actually, every sentence here has a great meaning, but we are going to learn this in medical entrance examination aspect. That's why it's enough actually. Alignment of sequences using computer-based programs. This requires supercomputers. Okay, genetic and physical maps on the genome were generated using information on polymorphism of the restriction endonuclease recognition sites and some repetitive DNA sequences. What do you say them as? Microsatellites, we see. The genetic and physical maps on the genome were generated using information on polymorphism of restriction endonuclease recognition sites and some repetitive DNA sequences. What do you say them as? Microsatellites. Okay, this is the area, the next area that along with the major approaches, this is also the very good area that means in examination point of view that we need to remember that 3164.7 million nucleotide bases, the calculations play much enough role in NEET entrance examination. The average gene consists of 3000 bases. Sizes vary greatly. The largest known human genome gene, dystrophin, an X chromosome, it contains that 2.4 million bases. Okay, the total number of genes are 30,000. 99.9% of the nucleotide bases are identical in all people, and 0.1% makes each of us unique. That means 1% to the other person, that 0.1% is going to be unique. With that only we are going to do the DNA fingerprinting, all the issues. The functions are unknown for 50% of the discovered genes itself. In the discovered genes, 50% of gene function we don't know till the time. And 2,968 genes are present on the chromosome 1, and Y is having the lowest number, that is 231 genes, Y chromosome. Less than 2% of the genome, that means nearly 1.5%, as we have seen in the previous video, that less than 2% of the genome codes for proteins. Repeated sequences make up very large portion of the human genome. These are going to play a very important role in fingerprinting. The repetitive sequences are stretches of DNA sequences that are repeated many times. They have no direct coding function, but they shed light on chromosome structure, dynamics, and evolution, or we say they shed light on DNA fingerprinting also. That about 1.4 million locations where single base DNA differences, we say them as SNPs, single nucleotide polymorphs, we say SNPs. SNPs are SNPs, single nucleotide polymorphs, occur in human being. That means in 1.4 million locations, the total sequence will be the same. Instead of 80, one base sequence, GC or TA, CG, will find over with a little change. What do you say? Single base DNA difference. What do you call them as that SNPs? Okay, so then let's continue with DNA fingerprinting. Then for the DNA fingerprinting, what we require actually repetitive sequences, and what we are searching for, that is very important for us. What we are searching for that in this particular area, DNA fingerprinting. So for that, I'll give a brief. Then after we'll go to the DNA fingerprinting. Repetitive sequences, what they are actually. We say them as genetic markers, the repetitive sequences. So first of all, let me show the repetitive sequences here. Then after only, and what exactly we are going to do in this particular area, that we need to know. That we need to know. Then after we'll get back here. Okay, so let me go for a brief explanation of that particular area. So I'm connecting to the board. Yeah, okay, you got the board. So yeah, okay. So what is this actually? That means we said already only 0.1% difference is there. In that 0.1% difference, repetitive sequences. Repetitive sequences are of various types. We are taking one example. For example, if I take ATTGAC, if I take this ATTGAC, I am writing here ATTGAC, not getting ATTGAC. So actually, I have some problem here. I'll go with whiteboard. I'll go with the whiteboard. Okay, so let me show here. Then now it's okay. I think, yeah. ATTGAC. This is a sequence. We say that this particular sequence again it will be repeated again. ATTGAC. I am showing only one side, single strand. I am showing that if you take one more ATTGAC in order, they are present like this. ATTGAC. So you can write any number of times here. Like if you see this particular repetitive sequence, this is a repetitive sequence of ATTGAC repeated for 4 times. We see. So this is called repetitive sequence that repeats again of 2 types. As I said yesterday, the clustered repeats. This is the clustered repeats. That means in a linear series, this particular repeated is going to be for many number of times. Okay. So what is the importance of these particular repetitive sequences in the DNA fingerprinting? That is very important. You need to know. So if a particular person is having these particular repetitive sequences, so not a rule like that. He got that particular whatever the repetitive sequences he is having. That all the sequences he should transfer to this particular the next generation to their children. Some of these will the particular child are going to get, not all. Some of these there is no coding, there is no importance, there is no function. But these repetitive sequences are going to be different from different people. If you take a child, he get some repetitive sequences from mother, some repetitive sequences from the father. Bigger science is, but we are taking a little bit, a small area. So let me take one more here. TAA, CG, TAA, CG, TAA, CG. Like this is the sequence from the mother. TAA, CG. So this is the sequence from the mother. So then I'm writing one more time, TAA, CG. Okay. So this is the sequence. So if this is father, if this is father, if you take this is the father one, and I'm writing here, this is any one of them. Let me say this is from mother, and this is father. So what can be the child? The child exactly this their particular child after the marriage of these two people. The child, not a rule like that. They he need to have the total number of sequences here. Little number it will match. Some he get from the mother, some he get from the father. He may get new even because it's a bigger changing area. So that's why we can say with these, we can say exactly every person may not have the complete similarity with the other person. This is a very important area. Then we call this as junk DNA actually. We say this as junk DNA. There is no use, but we are using now the forensic science for the DNA fingerprinting. There is no coding, there is no function, anything here. That as we said already, 30,000 genes, which occupy only 1.5%. And then after I said enhancers, promoters, regulatory sequences, that different kinds of microsatellite, mini satellites in the clustered repeats, these are all going to play a vital role here. So in that, I'm taking this particular area. If you take a full DNA, if you take a full DNA sequence, these particular repetitive sequences may present in one particular place, okay, one particular place. So what we do with them actually, by using the genetic marker, that specific restriction endonuclease enzyme we use, the another information you people need to know about the usage of restriction endonuclease enzymes. These are collected from the bacteria. these restriction endonuclease enzymes which are going to cut the particular DNA in specific places. Here, the molecular seizures, we say them as. Okay, so by using the restriction endonuclease enzymes, we are going to cut this particular in one specific place, in one particular place, and we collect these particular DNA fragments during this particular area. Okay, what we do, we analyze. Okay, so let's take a look here. Then after we'll get back. Let's take a look on this. The motive, I said, where we are searching for, what we are searching for. That we need to know. Then after we are going to start the method of DNA fingerprinting. The technique which is used to identify the similarities of the DNA fragments of two individuals. Alex J. Jeffrey identified this in the dystrophin gene. Alex J. Jeffrey identified this in dystrophin gene. Means the one more information you need to know that the dystrophin gene is for everyone it is going to be same, isn't it? That the protein dystrophin is going to make the integration of the muscles. Actually, those people who they have deficiency of dystrophin protein, they may get affected with DMD, Duchenne muscular dystrophy. Okay, so that you may get remembered over X-linked inheritance is that is present on the X chromosome. Okay, so the coding sequences, in between the coding sequences of the dystrophin gene, in between the coding sequences, that particular, in between the coding sequences, the dystrophin gene may have, is any problem? Is any problem? Any one of you? Just,
No, sir.
yeah, okay. Okay, so in case of dystrophin gene, let me write here. If this is the gene, so I'm going to draw a particular DNA sequence here. So if this is the dystrophin gene, I can't draw 1.4 million nucleated base pairs here, but let me see here. Like you may have some idea in molecular basis of inheritance. In between the exons, you'll find introns. That like introns, I am talking about repetitive sequences. This is a repetitive sequence which is present here. That means like A, T, T, ATT, GAC, that whatever present here. So this is the repetitive sequence. I am writing very short. So it will be repeated again. A, T, T. Okay, so that which I am doing with the red color, you have to understand that is actually dystrophin gene sequence. So again, gene sequence can see. Okay, simply we say that a movie when it comes in uh, television, a premiere, if a world premiere, you may find so many advertisements. For the completion of movie, it may take 6 hours of time. That because 10 minutes movie, 20 minutes ad, 10 minutes movie, 20 minutes ad. If you record that particular movie, how you see? 6 hours movie will come. That means for every, after 10 minutes, you'll find advertisements. So similar kind what I am talking. This is the area. Okay, so if this is the total dystrophin gene, in between the time, these particular repetitive sequences can be seen over. So dispersed repeats and clustered repeats. We say there are two types. No need. Just the only thing you need to know here. This particular area, dystrophin gene is same for you and me. But these particular repetitive sequences, number of repeats, the sequence of repeats may be different for you and me. These are all going to be maybe different for you and also for me. Okay, dystrophin gene is same for all. But in between the time, that means what I am saying here, if the same movie will come, now it came in Ma TV and it will come in G5, G Telugu. So same advertisements won't we get, isn't it? They may go with their own advertisements. When you play that in YouTube, they may go with their own advertisements. That's what I am talking. This area is different for all. Okay, gene is same for all, but ads, advertisements are coming in between. They are going to be different from all. So this area we are going to cut with molecular scissors, restriction endonuclease enzymes, and we are trying to find out this particular area. So that you need to notice. Okay. So okay, let me go for back here that PowerPoint presentation. Alex J. Jeffrey, that the technique which is used to identify the similarities of the DNA fragments of two individuals, developed by Alex J. Jeffrey. Okay, so Ishwar, you're getting the PowerPoint?
Yes, sir.
Yeah, okay. So the problem in my mobile, I think so. I'm watching actually in mobile. Okay, so that developed by Alex Jeffrey, 1985. The DNA of an organism carries some non-coding sequences called repetitive sequences, mini satellites. That's what I show now to you. What do you say them as? Variable number tandem repeats, VNTRs. These are all various types actually. For our knowledge, it's enough for us. They are repetitive sequences, they are satellite chromosome portions, and they are the VNTRs, variable number tandem repeats. These number of repeats is going to specific from person to person for your identification. I say again, these advertisements will come in between the movie. Movie is the dystrophin is going to be different in different persons. Okay, so what we do actually here, first of all, that we do isolation of the DNA from any cell or a blood stain or semen stains or hair roots, anything. If I rub on a table, my cells, stratified squamous epithelial cells will be on the table. So you can collect them also. So whatever it is, we collect that particular cell and we isolate the DNA. Then after that, what we do here, making many copies of DNA because that particular cell, I may not submit if I do a crime. So I don't go if I am the victim. No, if I am doing that particular crime, if I do that, I don't go again and again if I take my sample. So that's why we need to take copies of that. When you get a sample in the crime area, or when you get a sample, it is very important. The polymerase chain reaction, PCR. If you may remind over denaturation, annealing, and extinction. There are three here. That automated recycler that will within the short period of time, the crores of copies will get. That means simply we are taking a Xerox copy after the getting sample. They're taking the Xerox copy, multiple Xeroxes, PCR. Then after we do the digestion of DNA by restriction endonucleases, cutting them into pieces, in specific pieces. What do we do after that? Separation of the DNA fragments by gel electrophoresis. Gel electrophoresis we do. This is the diagram you can see here. The gel electrophoresis, when we do these particular fragments, will move into the different places based on their larger and smaller nature. The fragments are going into the different places. Okay. So next. Let me close. I got a problem. Okay, number 5. The treating with alkaline solution to denature the DNA. Denaturation. Do you remember that denaturation, the word we used in PCR also? What we do? We make break the bonds between the particular denaturing. You can see there that breaking the bond, making the double strand into two single strands. Denature DNA bonds in the gel into single strands. Then after we transfer this particular into the blotting paper of separated single-stranded DNA fragments to the synthetic membrane. It's generally nitrocellulose paper or a nylon membrane. And then baked in a vacuum oven at 80 degrees centigrade for 3 to 5 hours to fix the DNA fragment on the membrane. We bake them. Then doing is actually southern blotting technique. They place the nitrocellulose filter paper in a solution containing radioactive labeled single-stranded DNA probe. You need to understand what is exactly the probe. Okay, we made single-strand DNA fragments which are available on the nitrocellulose paper or nylon membrane. So do you know the sequence of that? Do you know the sequence? We don't know the sequence. So then what we do in the laboratory, we prepare DNA probes. Probe means all possible combinations like ATT, TAA, CGG, GCC, likewise, multiple possible combinations we prepare and we label them with radioactive coating. What do you call them as? Radioactive labeled single-stranded DNA probe. So this DNA probe binds with the complementary sequences of the DNA fragment on the membrane to form a hybridized DNA. That is to form a hybridized DNA. Okay, so that imagine if there are four fragments on the particular the nylon membrane, which is collected from the person. Okay, we don't know the sequences of that particular person, so that's why we prepare that lakhs of combinations in the laboratory and we label them with radioactive code. Okay, so in that lakh, when you send that all in nylon, go on the nylon membrane, actually this nylon membrane is going to be the dip into the solution of radioactive DNA probe containing area. So when we dip there, so the complementary radioactive labeled DNA will go and that bind with that single-stranded fragments, and that makes a hybridization of the DNA. The filter paper is washed to remove that unbound probe. As I said, over lakhs we prepare. Among them, some complementary will go. The hybridized DNA is then photographed onto an X-ray film by autoradiography. The image in the form of dark and light bands of the hybridized DNA obtained is called DNA fingerprint. This is called DNA fingerprint then. Okay, so then these are all said to be the DNA fingerprints here. That if you take that tandem repeats, as I said over, number of short tandem repeats. You see here on chromosome 7, that here the number of tandem repeats, and chromosome 2, number of tandem repeats, and chromosome 16. They cannot be homologous actually. That here you see that when it takes here that we got red color, just for example showing that red color, orange color, blue color likewise. Okay, so then when we DNA from individual A, okay, when you compare with the DNA from individual B, he is having his own sequences. You see here, he is having his own sequences. So it will come like this. So what we do with this particular area? What we do this? For example, if it all happened in a crime place, a hair is available. So we do that particular DNA fingerprint of that particular person. Will take it out, and those whom we are suspecting, they will try to match that particular fingerprints. Okay, or we say the parents, actually there is no permission individually. No person can go test nowadays. That forensic science is developed, but no child can go directly there, do it whether they are my parents or not. You can't do like this, but that is actually possible. That means that what I said to you, that complete repetitive sequences you may not get from your mother and father, but whatever they have, some of them you'll get, and you have additional again. So with that we can say whether they are parents or not. For the diagnostic of genetic diseases, to determine the phylogenetic status of animals, that we say these are all helpful for that, useful for the DNA fingerprinting advantages we can see. Okay, so before we just before we go for that, what I do